I would like to introduce the distinguished speaker for tonight, Naama Brenner. It's my pleasure to introduce. She's from Technion, the Israeli Institute of Technology. And so originally, like most of us, she studied physics and mathematics and also computer science at the Hebrew University in Jerusalem. She did her PhD at Technion. And then for a while, she moved to the US. So she worked as a postdoc at the NEC Research Institute in Princeton. She even worked for a while at a medical startup company. And later, she joined Technion, where she's now a faculty member. And so I met Naama, about 12 years ago for the first time, something like that. And she's always one of the most interesting people to talk to. Always has very, very new ideas and exciting ideas. And I think tonight she's going to talk about one of the topics that really interests her and that many people actually in our program are interested in is how is it possible that these small cells, of which all biological organisms are made to, how can they sense their environment? How can they learn about their environment? What kind of principles do they bring to bear to learn how to behave in their environments and to adapt in their environments? A lot of that is still very unclear, and it's almost like magic how things like bacteria can make a living in the most strange conditions and adapt to the most strange conditions, even when they're made on this small scale and are just little sacks of molecules. So this is one of the topics that Naama is very interested in, and she will tell you a bit about tonight.
Thank you.
I think I have to transfer this to you.
Yep. I have to do this somehow. Yep. Okay, maybe this is going to work. Can you hear me? All right, thanks a lot for the introduction, Eric, and thank you for having me here at this center for this public lecture tonight. I'm going to try to tell you a story about research that we've been doing at the Technion for a while. I named it The Brain Inside a Cell, Adaptation and Learning with Cellular Networks. And in particular, I'm going to tell you about gene networks. So here's my plan for today. The way I want to set the talk up is first I want to tell you a little bit what are gene regulatory networks, which appear at the title of the talk. And those are going to be, we're going to use analogies from engineering and from other scientific disciplines to try to understand what is a gene regulatory network. But then a most important question that I want to address next is how are they different from mechanical or artificial networks. And then I'm going to talk about how we bring to the lab questions about the properties of these networks, how we construct experiments and create models, mathematical models, to try to answer the question. And maybe I'll try to convince you at the end that really cells are much smarter than we might think, and that they do learn in some sense at least, in some primitive sense. Okay, so let me start by the first part in what is a gene regulatory network. So what is inside a cell? You might remember from your high school studies that a cell has a membrane, right, that defines its volume from the external world. It has this soup of molecules that's called the cytoplasm inside the membrane. It has a nucleus. Most cells at least have, a lot of cells have a nucleus. Some cells don't have a nucleus. Bacterial cells, which a lot of my colleagues are working on, actually don't have nucleus. But our body cells, for example, have nucleus, and they have a nuclear membrane. And what's inside the nucleus? Everybody knows, right? What's inside the nucleus? Yeah, DNA, right. Okay, so inside the nucleus is DNA, the genetic material. And that actually is the material that, in many respects, determines the properties of the cell and the properties of the organism. And what is DNA made of? And DNA is made of genes. And what exactly is a gene? All right, so we have this large molecule, the famous double helix structure. It's made from 4 types of nucleotides, and that holds in it genes which are pieces of DNA that are templates for coding particular proteins. Okay, just to set the just to be on the same page, proteins are these molecules that we think of as determining a lot of what the cells do. It's sometimes said that DNA gets all the glory, but proteins do all the work. So they're the ones that actually are the machines inside the cell. They determine a lot of the properties, and they are translated from these templates on the DNA into the molecules, and it's usually thought that every gene codes for one protein. So the history of genetics is very interesting. I'm not going to go into it. People have thought about the idea of genes even before the discovery of DNA. But really, the discovery of this one molecule that looks so much like a code book was a tremendously influential event in the history of biology. Because it was so crisp and so neat, and people thought for very many years that the notion was that really this is a code book, and if we only could read the code, we would understand all secrets of life, right? This was the promise of the genetic revolution, and that influenced a lot of the development of biology in the coming years. And in the spirit of an effort, a collective effort to try to decipher this code book, in 1990, which is several decades after the discovery, but many things happened in between. In 1990, several government agencies pulled together to make this big project whose goal was to read the DNA in the human body, to store the data, to develop tools to analyze the data. And really the feeling was that we're on the verge of discovering all mysteries of life. And what did I do wrong? Okay. So one thing, for example, that you could ask after starting, when sequences started to come in from the Genome Project, not only of human body, but also of other organisms, you might have asked, how many genes do we have? And one of the interesting things that happened is that before the Human Genome Project started, it was estimated that we have approximately 100,000 genes. As the project went along, this estimate decreased. And it was finally estimated that humans have approximately, now we know, approximately 20,000 genes, 20,000 different code words in our DNA. And this could be compared with the data of the Human Genome Project, for example, to a fly, which was a favorite organism of study in genetics. So the fly has approximately 14,000 genes, which makes sense. It's a less complex organism, right? So it has a little bit less genes. And then came the worm, and the worm has the same number of genes as we do. So what does that mean? Okay, so, and further, the project continued, and then the sequence of the DNA of rice was, can anybody guess how many genes the rice has? It has 50,000 genes, right? More than twice the human genome. Okay, so the clear conclusion from this is that the number of genes is not really an important parameter. It does not tell the whole story. And if we want this comparative study of genomes between organisms to tell us something about organism complexity, we have to look elsewhere. And actually, even before the Genome Project, the idea that it is not the number of genes, but something different, came along, and maybe it's the actual genetic code itself. And as early as 1975, it was already found that actually no, the answer is not there either, because, for example, between chimpanzees and humans, which have segregated more than 5 million years ago and are considered to be of significant difference both in lifestyle and physiology, actually the difference in the code words of their genetic code is really small. It's less than 1%. So that means that the code words cannot tell the whole story either. Okay? So we have to go back for a second and ask ourselves, maybe it's not, so it's not the number of code words, it's not the content of the code words themselves. Maybe it's something about how these genes work. Okay, so let's go back and look at the cell. This is a little bit more complicated picture, but I'm not going to go into details. I'm just going to tell you the following. The code words in the DNA have to be translated and eventually turned into a protein, a different molecule. And this process has many steps and is rather complex. So there's an intermediate molecule, and then it has to go out of the nucleus, and then it had to be read. And finally, after many steps, there is a protein. Okay? But who carries out all these steps? Right? Who are the workers of the cell? Who carries out everything that makes DNA into proteins? In part, it's the proteins themselves. Right? It's the proteins themselves. So once you created a protein, that can go back and influence the way that the code is read. Okay? So rather than having a linear process of a code and reading a code and something happening, you immediately get something which is circular. Okay? So the process, although the DNA looks really simple, it's made of 4 different letters and it's just a code book, the process of making proteins from it is very complex and is circular. So this is what we think of as a network. Okay? So the network symbolizes the fact that different proteins, which are the little dots here, affect one another in a circular way. So one protein can affect one gene, and that gene is transformed, and then it affects the first one back. Okay? And the connections between the different genes or the different proteins can be thought of as a network. Okay? And this network, this is the gene regulatory networks, and it controls what we call gene expression. So the word expression comes to highlight the fact that there's a potential in the DNA, but it's not necessarily expressed. It's not necessarily carried out. Sometimes the template is brought to bear, is turned into a protein. Sometimes it's not. Okay? You're free to ask questions, by the way, and interrupt me if anything's...
How many different proteins are there?
How many different proteins are there in each cell?
No.
Okay, so the different proteins are actually more or less the same as the number of genes that I was referring to before. Okay, so roughly, if each gene codes to one protein, then if we have 20,000 genes, we have approximately 20,000 proteins. This is the order, roughly. Okay, and the number, of course, varies between organisms. But the interesting thing is that now you have this genetic regulatory network, works like a switch system. So think about the cells in our body for a second. All of them have exactly the same DNA, right? We were born with a particular DNA, and that's all the cells in our body have the same DNA. But they're different cell types. For example, blood cells, right? They're these little round things, have a very different shape from neural cells. Neural cells have small bodies and large branches and help them connect to one another in the brain. So each cell type and skin types, they form layers. Each cell type has completely different properties. And remember, we said that those are the proteins which give the cells their properties. So although all of the cells in our bodies have the same DNA, this potential is brought to bear differently in each cell type. So this switchboard that turns some of the genes on and some of them off can have many, many different combinations, as you can imagine. And these combinations turn into the approximately 250 cells in our body. Actually, it's quite surprising that there are so few cell types, right? If we have 20,000 genes, you might have imagined there's like millions and millions of possibilities. But no, not all possibilities are actually cell types. We have several hundred cell types. But still, it is the gene regulatory network that controls these cell types, determines by the switchboard, the control switchboard, which cell type is going to be, which properties are going to be found for every cell type. Okay, so this is now, they say that every generation uses analogies from the technology of their own times to describe living things. And that's actually quite true. And many scientists have found that this kind of engineering diagram is very useful to describe a gene regulatory network. So this is a diagram describing the development network of a particular organism. I'll show you in a minute which one it is. And it shows with these arrows which gene affects which other gene, and which gene product, which is a protein, affects which other gene. And you could have looked at it and thought, well, that's like a control system of some factory or something like that. And there are analogies. This analogy is quite useful, and this actually describes the development of a sea urchin, which is a very nice and favorite organism for the study of development. Okay, but as I promised you before, one of the things that are most interesting, I think, to study is what is the difference between, what is the difference between the gene regulatory network and a factory switchboard, right? There's more to the living network than there is to the synthetic one. And to try to answer that question, let me put on a short quiz, okay? So gene regulatory networks, they control the development and the distribution of cell types during embryonic development. And what you see here, this is a famous figure from a very long time ago, showing 6 different embryos at the very start of their development. One of them is human. Can you tell which one it is? One of them is fish. One of them is salamander. They're very different animals. But at this very early stage of embryonic development, they're very similar to one another. As development proceeds, the differences start to show up. Okay? And maybe now you can see, or later at the end, you can tell that this is the human embryo, this is the fish, this is the salamander, turtle, etc. Okay? But notice that at the very first stages, they all look very much alike. Why does this happen? Why does this phenomenon occur? So the interpretation that people give to this phenomenon is the following. For each one of the organisms, development is controlled by a gene regulatory network. But all of these possibly had common ancestors. They had a common history. And they might have developed from one another through evolution. Okay, so the way we think about it is that this picture of the similar embryos at the first stages, that represents the fact that over long time scales of evolution, there were, it's like a funnel. There was a time where systems were very similar to one another, but at some point they diverged from one another. And this tells us that regulatory networks, in contrast to factory switchboards, right, they are modified by evolution. They themselves change as time goes by. Okay? So how do they change? How do regulatory networks change along evolution? And this is something that has been studied a lot. So, for example, not only different organisms, but even in similar species, body plans can change. And these are, again, determined by gene regulatory networks. And in the 1980s, a lot of the molecular biology of developmental pathways was worked out. And there were people used comparative studies between different, for example, butterflies to understand how the gene regulatory network of these different butterflies results in the different patterns, right? Once you have the molecular tools, you can compare the networks gene by gene and connection by connection, and you can figure out how they differ, how they develop from, how they diverge from one another. And there was two principles that emerged at the time. One of them was quite surprising, or maybe both of them were quite surprising. And that is that the elements of the regulatory network, the gene network, are conserved. What does it mean, conserved? It means that when you compare different species, the same molecules, the same proteins appear over and over and over again. So how does diversification occur? It occurs by reconnecting, by rewiring this network. So the picture that emerged from these comparative studies is that it's like a box of pieces, of Lego pieces. The way that the networks change is not by making up new pieces, but by reconnecting again and again in new ways the same pieces. Okay, so in some sense you have mix and match of the components, and if you think about it, if you have not even a huge number, but if you have a large enough number of components, you can make endless forms. You can make endless spectrum of shapes and patterns, and you can connect them in many different ways. So the logic of the regulatory system is what changes along evolution. Is that clear? Nobody has a question? Okay, I had a question at this point, and here's my question. So if we can understand the logic of the regulatory network like a switchboard of a factory, how can you mix and match elements of the logic of a switchboard? I work in the chemical engineering department, and if I ask my colleagues, what would happen to a factory if I took the elements and tried to rewire them and connect them in a new way? They would say, no, that's not going to work, right? That's not going to survive. It's not going to function. The system would collapse. So a very fundamental question is, so we know molecularly, we see that the elements are the same and that they're connected differently, but how can that happen? How can something reconnect itself, tamper with the logic of this regulatory system, and still the cell would survive? Why would the cell not die or collapse? Okay, why would it find a new way of functioning? And this question actually is related to a completely different question, it's related to a question, an old question in evolution. And that question is evolution fast or slow? How fast does evolution proceed? And Darwin, who of course didn't know anything about genes or DNA, but obviously knew something about evolution, he thought, and many scientists the same, thought that evolution must be a gradual and slow process. Otherwise, the cells would collapse or the organisms would collapse. Same argument as the engineers about this control system. So otherwise, if you give the organism too big a shock, it would not survive. So, okay, so Darwin says this, the engineers say this, but still we have evidence that this is how gene regulatory networks evolve. So what do we do? Okay, so obviously we're not going to answer this question by viewing the butterfly wings and seeing the natural world around us. Why? Because all we get to see is a snapshot of the current state of affairs. What I would like to understand is what happened when I tamper with the switchboard, when I rewire the network? What happens that enables the system to survive such a shock? Okay, and if I can answer that, maybe I will understand that indeed evolution can happen more rapidly than I might naively think. So we're here at the third part, which is how we bring, so there's no way to answer this by looking at the natural world. We have to do what physicists know how to do, is bring this into the lab. Okay? So at this point, our idea was, okay, let's think about a way to bring evolution under our microscopes to the lab and try to follow in real time what happens after such a change, such a rewiring of the network. The problem is that this happens once in a blue moon. This happens very rarely in evolution, and we weren't able to wait until this happens and then see. So what we did is there's several steps to bring. First of all, we chose a model organism that I'll tell you about in a minute, but the main ingredient is that by now technology has advanced to a point where I can induce such a rewiring myself, synthetically. Okay? Current technology in DNA basically allows you to cut or mix and match, like we said before, cut and paste DNA any way that I want. Okay, so the idea of these experiments was, why don't we induce or mimic evolution in the lab in a synthetic way and then follow the consequences in real time? Okay, is that clear? All right, and so we chose as our organism, as our model organism, the budding yeast, which we all know from pizza and beer, right? Beloved organism. It's a single-celled organism. It reproduces by budding. That's why it's called the budding yeast. This is the mother cell, and this is the bud. It has a short generation time or division time, so I can follow many, many division events. And because it's a biological model organisms, we know really a lot about its genetics, and we have a lot of tools to do experiments with it. So this is the experiment that we did, and in some way we tried to mimic this figure of the diversification of networks that I showed before in evolution. So this is our caricature of diversifying regulatory networks. We take the original network, and then we use technologies to sort of cut and paste the elements, the same elements from the yeast genome, and we can, in principle, we can get very different kinds of logical structures. All right, and now we have them in our lab, and we can see, is the cell going to die because of this tampering with this logic, or is it going to find some way to survive? And from that, we hope we might learn something about, in general, how under the natural occurring rewirings in evolution, how new life forms evolve and how novelty actually occurs. Okay, this is a large, I know this is an ambitious goal, but this was the thought when we did this experiment. Yes, thank you.
Are the regulatory links
Yes.
on the last slide
Mhm.
the things within the cell you showed at the first slide but didn't discuss in detail?
Yes, these are the okay, thank you for that question. So these links are now representing the effect of one protein on another. Remember that the genes in the DNA have to be translated or transformed into a protein, and in doing that, other proteins are involved. So that is what represents the effect of one protein on another. And these are these arrows, and I have, as I said, I have the technology to change them as I wish. And this is what we do. So let me give you just up front, before you get too tired, I'll give you the research question and the answer. And then we can go on and see the details of the answer. So the first research question that we posed was, first of all, can they survive at all? Can the cells survive under this huge perturbation or this really big hammer on the head that we're putting on them? Or in other words, was Darwin's argument about slow and gradual evolution, was that correct? If it wasn't correct, if they can survive, how do they do it? Can we monitor, can we look in real time and see what in the cell, what in the nature of the cell helps it to cope with such a change? And here are the answers, just as a preview. The answer is yes, they can adapt rapidly. We can see that. And the second half of the answer, how do they do that? And in general term, the answer is that this network that I drew for you before is really very flexible. It has a lot of ways to modify itself. And that is something which is different from the factory switchboard, and we'll discuss that when we get there, okay? Okay, so here's the result of the experiment. I'm taking one rewired network, and I'm showing you what happens. First, let's just look at normal yeast cells. So yeast cells, I showed you, they bud, they grow. This is what they know how to do. They know how to reproduce and grow. They grow little colonies. So if I put them on a petri dish in the lab, after a while I see these little colonies coming up, more or less similar to one another. More or less 2 days it takes them to grow to this visible size. So these are normal yeast cells under normal conditions. Okay, now I do the rewiring thing, and I put them in a situation which is conflicting and frustration. Okay, so again, I mimic what happens to them in evolution. What I think happens to them in evolution is that this mix and match of components completely ruins the logic of this system. And then after 2 days, I see practically nothing. Yeah, I don't see anything. And if I wasn't patient enough, I might have thought that they actually die. But if you give them enough time, after a while, 14 days, much, much longer than the regular yeast cells, they start showing up these weird-looking colonies. They look different. They take a lot of time. It looks like they're struggling, right? They're struggling. They're taking their time. Each colony figures it out differently at a different time. But in the end, if you wait long enough and you give them enough time, most of them will survive. Will find a way to overcome this rewiring of their network that I did. Okay, this is fine. Okay, so the first part of the answer is yes. I can see it in the lab. I can see that I'm not killing them by this perturbation. But how do they do it, right? This is the thing that we want to figure out. And to see how they do that, again, I have to use a different technology. Yeah, question, thanks.
So basically, with the yeast on the first plate, with the yeast on the second plate, the mutation of the yeast on the
Oh, no, they weren't. That's a great question.
Couldn't hear the question.
What was the question?
Okay, the question was, are these yeast cells a mutation? Oh, actually, you know what? Yes, you're right, because I misunderstood. Here's the question. The question was, are these yeast cells a mutation of these yeast cells? And actually, they are, because I induced the mutation, okay?
Mutation?
Yeah, it is because I cut and pasted the elements of the genome. In some sense, it's like a mutation, okay? It's not a point mutation, but it's like a reorganization, a genetic reorganization. So in a sense, it is like a mutation, yeah. Yeah.
Did you choose the mutation that you induced carefully, or was it random?
Okay, that's a great question too. So of course there's one choice here, but we did do several different kinds of rewirings, and all the results I'm going to tell you about are very insensitive to. This is practically arbitrary. Okay, it's practically arbitrary. It's not a very carefully chosen mutation. Otherwise, it would be a little bit not interesting, right? Yeah. Another question.
What was the mechanical means of perturbation? Electricity, or how did you actually rewire?
Okay, there are enzymes. There are enzymes that cut and paste the genome. This is a biotechnological. It's not a physical perturbation. It's a biotechnological technique that's well developed by now, and it relies on the sequence of the genome. It can cut the genome at particular places and paste it in other places. Today, a genome is really like a Lego. You can buy. You can design your own cut and paste of the genome and buy the appropriate enzymes, and you can do this with biotechnology. All right, other questions? Okay, so to understand how this is done, you have to look inside the cell, right? It's not enough to see them grow. To see them grow is enough to say that they're succeeding. But now I'm going to go to another technology, and this is the microarray technology. It's actually not that new. There are newer technologies by now, but this one is quite good because it spreads the material of the cells on a chip, okay? And using the properties of DNA, it shows you which genes are turned on and which genes are turned off, okay? And because the budding yeast is a model organism, the companies have made chips that specifically include the genome of the yeast cells. And this enables me to take the yeast from my experiment, spread them out on this chip, and then read optically, right, to get a list of the genes. This one is on, this one is off, this one's high, this one's low. I can know that for every single gene in my yeast genome, okay? And what we saw when we looked in those chips is that while the yeast cells were struggling to start growing on our plate, their genes were starting to go wild. The genes were starting to express up and down, different levels, something very unlogical. Yeah, question.
So when you do something to change the wiring network, does the actual DNA in the gene change as
Yeah.
well?
Well,
Or do just different genes start to become more active?
no, it's the structure, but as I showed before, that happens in evolution. It's not the genes themselves that are changed, it's the way they're connected to one another.
Okay.
All right? It's not the elements, but it's the connections between them that are changed. But the way that the genes are expressed This is something that I have no control over. This is something that I'm measuring now. And I'm telling you that the result is that they start to go wild. They go do a lot of up and downs. Every gene does something different, and they really respond as a collective, and it looks like they're kind of looking around for something. Yeah?
Could you explain that figure a little more? What are the dimensions? Is it time?
Oh, no, this is the way that this is constructed is you take the DNA of the yeast and you kind of spread it out on a chip. So every point here might be, this is kind of schematic, right? This is not a real experiment, but every point here might be a gene or a piece, a segment of the DNA. And using the hybridization properties of DNA, you take your own cells and you kind of spread them on, and then you can see the material from the cell. You take the material from the cell and you can see which genes have been, yeah.
So each circle represents the growth of a single gene. Or the
Yeah,
expression.
more or less. Yeah, exactly. So the space here, it doesn't have a physical meaning. It's just different locations in the genome.
So there's about 300 genes or something?
Yeah, this is not a real experiment. This is just an illustration. Okay, here I'll show you an experiment. I'll show you an experimental result. Okay, this is the first real experimental result that I'm showing. Okay, so I told you that the genes start to do unexpected things in this experiment, and as physicists, we're brought up to say, when you want to trust your experiment, you have to repeat it many times and get the same result, right? This is good science. You have to do the experiment again, see what happens. So now I'm showing you actual data. This is actual measurement. And what you're seeing here is the following. This is experiment number 1. This is experiment number 2. So I did the entire thing from the start. I started with a new yeast cell. I rewired it again. I grew it again. I measured the genes again. Okay? So 2 different separate experiments. Every point here represents a gene, 1 gene. And the axes here represent whether that gene was turned on or turned off. Okay. If I would expect the 2 experiments to do exactly the same, I would expect, this is experiment 1, this is 2, I would expect the dots to look like this, right? Because whatever was low in experiment 1 is low in experiment 2. Whatever is high in experiment 1 is high in experiment 2. That's not what we got. We got the very intriguing and, in some sense, troubling result that every gene does not repeat its actions in the next experiment. So the genes, one thing is go up and down, and secondly, they don't do the same thing every time. All right, and this is not an error of the measurement. We know how to control for the measurement errors to say that this is real changes from one experiment to the next. Okay, our interpretation of this was the following. There's something improvisational going on, right? Something is that the cell is doing something which is a little bit random. It's trying out different states. It's exploring its options until maybe at some point it was lucky enough to find a solution. Okay? So different genes do different things each time. The cell adapts by some kind of improvisation. This was our interpretation. Okay. So how do we know if this is possible? All right, let me tell you about, right. How do we know, how do we get, like I said before, every generation takes its analogies, right, from the current technology. So I want to try to tell you how we thought about this problem in terms of some other technology that's very well known in our times. And this is neural networks. So let me tell you something about how artificial neural networks learn. In artificial neural networks, now the hottest thing is, of course, deep learning, deep networks. But the way this works is that they have many connections in them, right? Like similar maybe to the gene regulatory networks. And these connections can be modified. They can change. They can change from strong to weak, and they can be modified. And to teach a network a particular task, basically all that you're doing is changing the strength of the connections between the nodes. This is how artificial neural networks work. And now, everybody's heard about the AI revolution. And by now, we know that artificial neural networks can easily beat the best chess players and the Go players and whatever. they do many, many sophisticated things. And all of this is done by just changing the strength of the connections between the members of the network. And so maybe also in gene regulatory networks, something like that can be happening, right? And as you start to look into the mechanisms of the biology behind these networks, you realize that the network connections are actually not that rigid, right? There are many mechanisms in biology that modify these connections, makes them stronger or weaker, make them context dependent, and this is especially true under stress. So if what we did to the cell caused them to be under stress, maybe what they're doing is trying to modify their own connections in an attempt to find a solution. Okay? So we don't know how to address this experimentally yet, but what we do know is to construct a mathematical model and try to figure out how this works. And let me try to explain to you in a couple of sentences why this is a mathematically or theoretically interesting question. So we call this the hot-cold random search. So in Israel, we have this game for children, maybe in America also, where you hide something in the room and the child goes around, and then you only give it a hint whether it's cold or hot, cold or hot. And then slowly, gradually, it finds it, goes around and searches and explores the room until it gets to the point and finds it. And that usually doesn't take very long, right? But this room is two-dimensional. There's only so many places that it can go. Think about our gene regulatory networks. It has thousands of genes in it. How on earth am I going to find the right configuration with each gene, exploring around like a child in a room? There's no way I'm going to, it's not trivial, right? It's an interesting question. Under what circumstances can this find the right configuration? And so here's the basic model that we constructed. So this is the technical way that we drew it in the paper, but you can look at it this way. So here's stress. This cell is under stress, and when it's under stress, it keeps searching around. It keeps changing its gene expression levels randomly, if you like, and it goes on and on and on until at some point stress is decreased. When stress is decreased, it's happy again, and the level of changes are decreased as well. Yeah, question.
Is your inconsistency in experiment 1 and experiment 2, is that characteristic of the new organism, which are unmodified yeast? Do the same thing?
No, it would not be that different, right?
It's not under stress.
Yeah, that's some control that you have to compare to, definitely. Yeah, it would not be as different. Okay, so this is the model, and we implemented it in a computer program because it's a really complicated model system. And here's the result that we found. We found, first of all, that sometimes this works. Sometimes this game of hot and cold with thousands of degrees of freedom can work and find the right solution. However, some networks are better than others at this thing. So for example, if you took just a random, simply connected network, that would do really bad. But if you took a structured network, see these structures, that there are these large hubs in this network that are connected to many others. So this network has some structures in it. It has some non-regularities, irregularities. This kind of network performs much better at this type of hot and cold game than the random ones. And interestingly enough, the gene regulatory network is nothing like a random network. It actually has such hubs that are called master regulators and are known in biology. And we thought maybe this could be something that's promoting the adaptability of these networks to change. So the analogy is at a very top level, but let me just say it anyway. You may know that until about 8 years ago or 7 years ago, people thought that artificial intelligence is a dying field. And then in 2012, something happened that changed that. And that something is related to the structure of the network. That was the revolution of deep learning. And the revolution was that people, among other things, one of the things was that people discovered that the structure of the network matters. If you have a lot of layers if you have a deep network with a lot of layers, that can do many, many more things than just a shallow network. Naively, you might not have thought that such a difference would be important. But it's the same with genetic networks. A structured network can do things that a random network can't do. And unfortunately for us physicists, we like to think of things as being universal, simple, random. Unfortunately for us, we have found this, and I think this might be a good lesson for us physicists who are working in biology, is that sometimes details matter, sometimes structure matters, sometimes it's not the most simple random model or picture that gives you the answer. So this is as far as our analogy goes with the neural networks. And let me now summarize what we've learned. So first of all, before I summarize, I have to say a disclaimer. I've given you a combination of textbook material, right, and things that are known to all, and experimental results, and I allowed myself to go all the way to like kind of wild speculations that are obviously not well established yet and probably, you know, under debate with a lot of other scientists. But here's the picture that's starting to emerge from what I showed you. So as in the famous song, everything is changing, and the real difference between regulatory networks and mechanical or artificial networks is that they change. There's a hierarchy of changes. So what we've seen is the following. First of all, they change in evolution because their elements mix and match naturally by mutations. And this is one of the important driving forces in evolution and in speciation and creation of novel forms. And secondly, we have found that really evolution can happen quite rapidly and quite dramatically because the structure of these networks allows them to adapt to these changes. Okay, so we have multiple levels of layers of change, and the time scales are different. This is a time scale of evolution. This can happen in principle within the lifetime of one yeast cell, and so short that I can see it recovering and adapting in my laboratory. And again, like in the song, different levels, also the superficial and the profound, the different levels are changing simultaneously on different times. Okay, so just to close this seminar, let me say that one of the joys of doing science really is working with good friends and making good friends. And let me just introduce you to the main players in the things that I've shown you. So the experimental part was led by Eris Brown, a brilliant biophysicist at the Technion, who is a great friend and really led the experimental effort. The exploratory adaptation ideas, models, and other parts were done in collaboration with Yoav and from the Weizmann Institute. And the concept of cellular learning, which we are now developing further, is done in collaboration with Omri Barak, who is actually a neuroscientist in origin, but is now being convinced to think about these learning processes in a very much more broader sense. And I think cells should not be underestimated relative to brains. There's a lot of things that they can do. Thank you for your attention. That's it. Yeah.
What are the stresses that you referenced in your diagram
Okay.
in real life that cause cell configuration?
So you want to know more about what is the cellular stress. So there are many ways for a cell to feel its own stress level. Let me give you an example. The proteins that are being made from DNA have to fold into the correct shape. Okay? If there's stress in the system and conditions are not ideal, not all of them will fold into the right shape. The cell can sense the level of misfolded proteins inside itself. And there are many ways that it responds to that. Okay? There are even known mechanisms that it responds. For example, there are special proteins that refold the unfolded proteins, but at some point maybe there are too many of them. So that's one way, for example, of sensing stress. And this is very global. It has to do with all the proteins. It's not very specific. If conditions are bad, proteins will have a hard time folding. But that's just one example. There are more examples like that. Yeah.
So, how do you test your model?
Oh, great question.
For example, you've got a great model, it seems consistent with the biological data.
Yeah.
But can you make predictions
Right.
from your network model and then go back to the biology and test that?
So currently I can't. We're actually working and we're thinking of developing the next generation of experiments, because these experiments are quite old and the technology has advanced a lot since 2007, whatever. So yeah, we're thinking in that direction. Currently, the model is only conceptual. Unfortunately, I don't have any, predictions. Yeah.
If you have a neural network, the number of ports available is important. When you get to the biological system, how many ports or contact points are there per cell? Or
There are several thousand. There are several thousand. In yeast cell, there are several thousand. That should be enough. There's a lot of combinations you can.
And when they connect, are some of those ports that must be left open? So there
Some of them are connected to the external world, yeah. Some of them are input kind of, yeah.
some are just left
But
open.
this is, there's an important difference between most artificial neural networks and our network, and that is exactly that usually artificial neural networks are what's called feed-forward. So the architecture of feed-forward means that you have a signal coming in from one side, then it goes through something, and then it comes out the other side. Our architecture in biology, these are recurrent networks. They're recurrent, they're kind of closed on themselves. So that's one difference between the two. But in learning theory, people are thinking also about recurrent networks. So we know a lot about those as well. Yeah.
What would be the ultimate fate of a modified cell? Do they potentially live forever
Okay.
if they have the right environment?
I mean, what we've seen in the experiments, maybe surprisingly, is that if you let them adapt to the stress, they can do better in some senses than the original yeast. Yeah, they can do better in the original. I could explain the details, but it's not that important. They could, for example, survive in chemical agents in higher concentrations than the original yeast cells. So, yeah.
Can you relate the development of antibiotic resistance to
Oh,
this?
yeah, beautiful. Thank you for that question. So we are also thinking about extensions. We have one implementation of this, and there are several other experiments of the same kind on the yeast cells. But we think there are other examples in nature where things are happening in a similar way, and drug resistance is one of them. Okay, we think that cells that are developing drug resistance, there are different ways to develop drug resistance. Some antibiotic resistance are developed by just a point mutation. That's a different process. But sometimes cells can develop drug resistance by a similar, we think, similar learning process. Yeah.
Network.
Yeah.
How would, like we hear about targeted medication or targeted treatment based on your genetic content, but it sounds like this system, if it's constantly changing, how could you, that puts a damper on the idea
Right.
of personalized medicine targeted
Absolutely.
genes.
Yep. Absolutely. I agree. I would go even further, even a step back, and say that even the notion that you can trace diseases back to particular genes is recently becoming quite questionable. There's a very small fraction of diseases that can be connected one-to-one to a particular gene. A lot of the diseases are turning out to be influenced by hundreds of genes, and each of them is a tiny contribution, and it's a network effect. That's, yeah.
Back to one of your earlier slides when you're talking about how many genes are in organisms. Is it because that as you get to be a larger organism, your proteins get more sophisticated, so you have fewer larger proteins?
No, actually not larger, but more. Yeah. Yeah.
But you have fewer proteins as you become, what's the difference between a human and a rice?
Yeah.
By a factor of 2.5.
Okay, so that's little bit of a complex question because it depends what kind of protein. Some of them increase in number, some of them don't. The kind of division of kinds changes. I don't know if I answered your question. Do you want to answer? Yeah. Back.
You, I think, indicated that you work in a chemical engineering.
I do.
You talk of physics, you talk about this is a biological discussion. How important is the multidisciplinary nature of your colleagues to the evolution of learning?
Okay, so I'm a member of the Network Biology Research Lab. These two guys over here are leading scientists in artificial learning. And as I said, my collaborator Omri is a neuroscientist who's working in learning. I think it's really important. We're kind of influencing one another. We're learning from one another. I think that's crucial. I do. Yeah.
I've taken a bunch of steps back. How would you define memory and learning at the cellular level?
So memory and learning, okay. Okay, learning in a sense is the ability for a system to reorganize and modify itself in order to be able to perform a new task, okay? So it has to do with change, internal change, and it has to do with the relation with the environment, because the environment requires or demands something from the system. It's something of a closed loop between the system and the environment. Memory is maybe even harder, but memory has to do with the persistence of the newly acquired state. I don't know if that answers. These are hard things to define, of course. Yeah.
When you modified the yeast, you had two samples.
Mm-hmm.
Have you introduced the two samples to each other to see if one would become
That's a
dominant
good question.
over the other within
No,
the sample?
that's a good question. We didn't do that. No. Any other? Yeah.
Have you or your colleagues done any work on biofilms, which seem to be self-organizing colonies and confer
Yeah.
resistance?
Yeah, that's a really good question. They kind of learn a new behavior when they turn into a biofilm. I personally haven't worked on that. I know some people who work on it, but that's a good point. That's also a different state of their, yeah. Yeah.
Could you give us a clue on what the actual change that you introduce it? You distinguish that it's not a point, you don't change the DNA.
Oh yeah, you change. Do you want me should I go into details? I don't know. I mean, I'll just do it schematically. So there's a gene, right, that makes a protein, right? That gene, the way that regulatory control works is that there are other proteins that bind here and start this process off. What we did is deleted all of this, right? And connected this to a different piece of DNA. Okay, now this is the logic of this gene. It reads the correct inputs, it translates that information into whether this gene needs to be expressed or not. We have ruined all of this logic by just detaching all of its inputs and just connecting it to something different. That's in principle.
Successive generations of the cells.
They inherit this.
Contains-
Yeah, this is a genetic change. Yeah. Thank you.